So, as you guys were going through the codon charts, or excuse me, the DNA template, like I say, the start codon was the third one, which should have been methionine, and you should work from there all the way to the stop codon and coded out your proteins, and it should look something like that. DNA, your mRNA. We're going to talk about tRNA here in a second. We're going to add that in, and then the protein or amino acid sequence that's constructed from there. Okay, so before we do that, though, let's finish up part 2 of the notes. Where do we get to? Oh, this is the hard part. The 3 phases of production of a transcription unit for making a piece of mRNA. So flip over to 340, yeah. All right, so as always, it's probably a little bit more complicated than what we're going over here. Let's just, I'm going to go through the notes here, but then we'll look at the book as we go to. So you have this promoter. The promoter helps to promote this to happen. It's usually a group of enzymes and proteins that are working together to help this transcription take place. You see RNA polymerase is in there. RNA polymerase sounds a lot like, yeah, except instead of making DNA, it's making RNA. So first thing that happens, the initiation. This is the building of the factory. It's called a transcription. The protein is called a transcription factor that attaches to the Tata box. So you may see this come up. Basically, along the DNA sequence, this RNA promoter, the set of enzymes, has to know where to attach to. And so it's looking for this DNA sequence. It shows it on 341 at the top right, the promoter. You see that Tata box where it's like TA, T, AA. So that is the infamous Tata box where the promoter attaches. And then there's a stop point right there at the end of that Tata box where the DNA will be copied. And there's a template strand. So that tells it which side is the template strand to be copied and which side is the non-template strand, which will not be copied. Okay? And so basically what happens, initiation, it grabs a hold of it, additional transcription factors are added, proteins and enzymes, and then RNA polymerase joins it. The RNA polymerase is the main one there that you need to be familiar with. In step 2, elongation. So the enzymes and proteins are in place, and they are in the right place, ready to make a copy of that DNA, of that gene. They're ready to make their mRNA. It has to go in the 5' direction. RNA polymerase separates the double helix. It adds in nucleosides. What are nucleosides? We talked about this the other day. You might listen to that lecture? Of course not. A nucleoside is a nucleotide plus 2 phosphates. What do the 2 phosphates do? Gives it energy. You have to have energy in order to make those covalent bonds between the nucleotides. So instead of having just a nucleotide where you had like adenine and deoxyribose and a phosphate, it throws on a couple more phosphates, and those are used in the bonding process. Okay? So you have to have nucleosides in order to make the mRNA, or in the case of DNA, in order to replicate DNA. Okay? Well, in this case, yeah, so you'd have to have the nucleoside that's made of, you could have one adenine and ribose is the sugar in this case, and then your phosphate plus the 2 other phosphates used for energy. Okay, elongation. Where do those nucleosides come from? That's a good question. Where do we get our nucleosides that help to make RNA? You're not born with them. Well, not most of them. Where do you get the DNA nucleosides to make more DNA when your cells divide?
Aren't they like just recycled?
Well, some of them, but like if you're little, then they're not just going to be recycled. You've got to have more, right? Food, yes, that is the correct answer. So in order to grow and develop and get bigger and get more of something, you have to bring it in. And so we get nucleosides from the food that we eat. So when you eat plants and you eat animals and they have DNA and RNA in there, we eat it and consume it and chop it up and then reuse it. Thank you, plants. Thank you, animals. Now, if you're eating pure sugar, that doesn't have a lot of that in it. It's just all sugar. So remember that. Make sure you're getting all your DNA in a healthy, well-balanced diet. All right. Where do we go? Lysosomes up to recycling, blah, blah, blah. Part D. After RNA polymerase 2 has passed the DNA transcription point, the DNA reforms the helix. So basically it snaps back together. Cells can make multiple copies of RNA because the DNA is left intact and protected. So it's able to make multiple copies as needed, come back as many times as it needs to because that main template is there. Termination, part 3. What does termination mean?
Stop.
It's the end of the world as we... Yes, it's the end. So, or stop point. So termination means it reaches that stop codon, or one of them, and stops, break loose, and is released from the DNA. Boom. There you go. All right. On 341... Here's a little bit different picture, basically the same thing. Remember, the promoter attaches the title box. The transcription factors start with elongation. What are those 3 big purple blobs on 341 in the middle? What does purple blob mean in biology book? Purple stands for protein. So that's showing you the proteins and enzymes that are being used in that transcription factor. And at the bottom, you see all of them added on there. You got the RNA polymerase added in. You're ready to go. All right, next page. 342. This shows you a picture of elongation. So as the DNA is unraveled and it's being, nucleotides are added into the mRNA. You can see how they're added on there. They're being added in the 5 to 3 direction. Dang, you're good. All right, 343. Modification of the primary transcript. We talked about this yesterday a little bit, so I'm not going to spend too much time on this. Modification of the mRNA molecule. A protective cap is added, like pretty hard. So all this is about protection of the mRNA. Eukaryotic cells have a ton of enzymes floating around in the cytoplasm, and so you don't want these destroyed before they get to where they're supposed to. At the backside of your mRNA, a poly A tail is added. Poly A means there are people in here, they're just not answering me. Many adenines. 50 to 250 adenines will be added onto the tail. Protects against digestive enzymes in the cytoplasm. So they're trying to protect that and maintain the integrity of the mRNA before it gets to the ribosome. Okay? I'm boring myself. It's okay. We'll live through it. All right. Seen worse. All right. Number 3, middle modification. This is where we have RNA alternative splicing. Ooh, that sounds complicated. DNA introns get removed in this process, so they're cut out using spliceosomes. Spliceosome is a fancy word. It's splice, you think, to cut something out, is a collection of SNRPs. Now, I'm going to collectively call this SNRPs because that sounds fun. These SNRPs are basically just scissors that are cutting the introns out. The exons are stitched together, and they will then be the final product that goes to the ribosome to code for a protein. You only see these introns and exons in what type of organisms? Eukaryotic organisms. Good. Remember prokaryotes, they're very efficient. They don't mess with all this. So, if you look down on part B, I think we talked about this yesterday, really important for the making of antibodies in our immune system. And so basically what it's saying here, and I'll give you the simple version, is different sections of those exons can be mixed up and matched in different ways so that they're making different proteins. So they're making different antibody structures that could attach to different types of viruses or bacteria. And having all of this variability in that protein structure is a really good thing. It diversifies our immune system. Okay. In part C, they get stitched back together, and then you're on your way. On 344 at the picture, it shows a picture of this spliceosome, the SNRPs there at the top, where they're being used to cut out those introns, and the exons are being stitched back together. I think that's about all you need to know from that one. Okay. Any questions? Super. Let's talk about tRNA, and then I'm going to let y'all finish up this little sheet y'all are working on. Part 2. And turn to page 345. What is tRNA? No, it is green inhalers on your book. Yes, it is transfer RNA. So what is it transferring, Katie?
The amino acids.
Okay, it's transferring amino acids to where?
The ribosome.
Ribosome, so that we can add them together and they're coded for. Now, what do you notice about the codes on the mRNA and the tRNA? On 345. Do what?
The tRNA is the code that the DNA had.
Almost. Minus
T's.
Yeah, so it's the what of the codon? Well, it calls it the anticodon, but it's really the complement, right? So in this case, if you have UUU, you have to match an anticodon up, which would be AAA. If you have GGC, then you've got to do CCG. So it has to match up with it. So on your sheet that you're doing, that's why I put tRNA, leave a little bit of space for it. You've written out your codons on there. I want you to put the anticodon that's going along with that. Now, before you do that, let me explain something. When you're coding a protein, you are only going to look at the codon and the codon chart, okay? The only reason we're doing this is so that I know that you understand that the anticodon has to match up and pair up with the codon in order to drop off its amino acid. But when you're actually coding for this stuff, the code comes from the codon, okay? Any questions? You got that, Katie? You're looking a little scared there. Okay, so you always, when you're coding for the amino acid sequence, you always look at the mRNA codon. Good. But tRNA has to match up with it in order to drop that payload off, so to speak, in order to make that protein. Okay? So let's take these last few minutes. I want y'all to finish up that sequence y'all are working on, figure out what anticodons would go along with it, and we will be done for the day.